Here we present our recent publication on automated classification of B-cell neoplasms using deep learning on multiparameter flow cytometry data. This work is published and is available online in Cytometry Part A. So let's begin. Cell, also known as B lymphocyte, is a type of white blood cell. These cells secrete antibodies and are part of the adaptive immune system. B cells develop from the hematopoietic stem cells or HSCs that originate in the bone marrow. These HSCs further differentiate into myeloid or lymphoid progenitor cells that further branch out into specific cell types. Our B cell originates from the lymphoid progenitor cells and develops in several stages. B cell divides and multiplies as necessary. Sometimes these cells multiply uncontrollably, giving rise to a malignant growth known as B-cell neoplasms. Neoplasms can be broadly and informally classified into Hodgkin's and non-Hodgkin's lymphoma. In our study, we focus on B-cells, non-Hodgkin lymphoma, as B-NHL hereafter. B-NHLs can be further classified into various subtypes that are shown here, some of which are more common than the others. And the identification of the right subtype is essential for the prognosis of the disease. An integral diagnosis and monitoring of such hematological disorders is known as flow cytometry. In flow cytometry, the cells are passed in a sequential manner through an instrument called flow cytometer. The flow cytometer illuminates the cell surface with a light source and captures the emitted radiance. There are two types of emissions. The first one is called as the forward scatter. This tells us information such as the size of the cell that can be used to differentiate various immune cells, for instance, monocytes and leukocytes. The second emission is called side scatter, and this gives us information about the granularity of the cell. In multiparameter flow cytometry, along with forward and side scatter, antibodies that are coupled with fluorochromes are added to the cell before passing them through the cytometer. Now, when the cells are illuminated, these antibodies that are coupled with fluorochromes emit a fluorescence at a specific wavelength and is measured by the device. The measured fluorescence can now be plotted in a 2D scatter plot that is shown here. For instance, our B cell that has no granularity and expresses CD45 would fall on the right lower corner of such a plot, whereas a different cell, let's say a monocyte that is far more granular and still expresses CD45, would fall in the upper middle part of this plot. By plotting these markers in a 2D space, one can differentiate different cell populations. To diagnose the correct subtype, an expert would have to manually gate such plots to identify the cell populations and to quantify these cell populations. This manual process of gating is very tedious and time-consuming. As the number of markers that are measured increases, the number of plots that needs to be analyzed increases exponentially. This process is not only very time-consuming, this is also a subjective process, as in the gates that are drawn varies between experts. If a machine can be taught how to read the flow cytometry data and produce these class predictions without any manual work, this process will be much faster and much more accurate, and this is the aim of our project. To accomplish this, we obtained blood or bone marrow samples from 20,000 plus patients. That includes 8 different types of B-NHLs and healthy cohorts. It was further divided into training and test data in a time-based manner to simulate routine diagnostic settings. The training data was then used to train a neural network that generated class predictions for all the 9 subtypes that are in our dataset. For each of the patients, up to 3 tubes of samples were acquired. Each of these tubes, 9 monoclonal antibodies are added. This is then passed through a Navier to acquire the data. So in our data set, 3 tubes are measured and in total 28 unique markers are measured. Now I will use a two-step process. In the first step, we generate a self-organizing map or a SOM. A SOM is a type of artificial neural network that performs unsupervised learning. In simpler terms, what a SOM does is it clusters the many data points into a smaller dimension based on the nearest neighbor approach, all the while preserving any spatial information that is encoded in the data. For instance, let's say data point X and Y are closer to each other compared to X and Z in the original space. The SOM would cluster these data points in such a way that in the reduced dimension, X and Y are still closer compared to X and Z. In our workflow, we use SOM not only for reducing dimension, but also to produce a sort of low-resolution 2D image of the raw flow cytometry data. Images generated for each of the tubes separately are then processed by a convolutional neural network or a CNN. The idea behind CNN is that there are multiple convolution layers and each convolution layer picks up a specific feature of the image. In our case, we process each of the images for each of the tubes separately in 3 convolution layers that picks up the features of these tubes. These are then concatenated and processed by 2 fully connected layers that produce the class predictions that we are looking for. Part of this process is a score that is assigned to every subtype that the network learns, in our case, the 8 classes. And the highest score is the likeliest diagnosis or the predicted class. Here you can see a confusion matrix. A confusion matrix shows how well the predicted labels coincide with the actual true labels. The scores in each of these squares are the F1 scores. F1 scores are harmonic mean between recall and precision and is a widely used measure in a multi-label classifier. The F1 scores here tells us how well a particular class was classified by the neural network. For instance, a score of 0.99 for CM indicates that 99% of samples that are actually CM were classified correctly by the neural network. By following our two-step approach, we were able to achieve an overall weighted F1 score of 0.94. That means 94% of the times our samples were correctly classified by our neural network. Now that we have a model that was trained on a huge dataset of 5,000 samples that performs on the same level as a human expert, the goal or the current work focuses on making these models more robust and adapt these models quicker so that they can be integrated in a routine diagnostic settings. What we mean by making this model robust? These models must be able to work with not only the base data that it was trained on, but data sets that are similar but slightly different to the ones and much smaller to the ones that these models were trained on and still achieve a good performance. This is the current work that we are working on, and if you're interested in knowing more about the project, contact us or refer to our publication.